Welcome to the afternoon sessions, everybody. My name is Matt Romosier. I'm a master's student in reproductive physiology here at Iowa State University. It's my pleasure to introduce our first speaker for the session this afternoon, Dr. Randy Prather. Dr. Prather is a distinguished professor in reproductive biotechnology in the Division of Animal Science at the University of Missouri, as well as the director of the NIH-funded National Swine Resource and Research Center. Dr. Prather's lab there in Missouri has made over 1,000 cloned pigs that represent over 45 different genetic modifications for agriculture and medicine. If you could, let's put our hands together for Dr. Randy Prather.
Thank you. I assume the mic's on, people can hear me. Okay, so I'm going to talk about genetically engineering pigs today. And you might not think of the University of Missouri and National Institutes of Health in the same sentence, but we've been able to use the National Institutes of Health funding to develop a lot of the technologies that I'm going to talk about. So the outline, first I'm going to give you a little background on genetic engineering. We're going to talk a little bit about CRISPRs, not very much, just barely introduce them. And then I'm going to talk about something near and dear to most of you, and that's porcine reproductive and respiratory syndrome virus. And I'm going to talk about two molecules that are thought to be involved, at least were thought to be involved, in pigs getting infected with PRRS. And those are Siglic-1, which is also siloadhesin, and CD163. And what I'd like to do is end up at the end with some applications. What other neat things can we do with this technology? So you guys may or may not be aware, but pigs are used quite extensively as models of human disease. And this is a partial list of where the pigs used. Cardiovascular, pigs really replicate what happens with the cardiovascular system well, but you can go through the list. Organ transplantation. Pigs as a source of organs for humans that need organs. That's one of the things that we're working on at the University of Missouri. And this is a partial list of the genetic modifications that we've made. I didn't list the gene name. I list the acronym for the gene because that fills up the slide anyway. Today, really what I'm going to focus in on is disease resistance. But as you look through this slide and kind of see some of these things, phenylketonuria, spinal muscular atrophy, muscular dystrophy, some of these other things, we've made pigs to address those issues because in many cases the pig is the model for studying these diseases, especially like cystic fibrosis. We have a number of other genetic modifications that are in the works, and that list is here. Most of these are biomeds. Some actually cross over and affect human medicine, so things to help us understand metabolism. Again, more modifications for cardiovascular disease. Organ transplant, we have a Prader-Willi syndrome model now. We've made the numbers now over 55 different genetic modifications to address human diseases and also agriculture. So before we make a genetic modification, we gather a bunch of information. Some of the information we gather is we take cells and we grow them in the lab, we get them sequenced. The group here at Iowa State's been involved in helping to assemble that genome so that we have a blueprint to understand what really is controlling development of the pig. We then look at, well, what about other species? What about the human? What about the mouse? How are they different? How are they the same? And if we want to replicate a condition in the human, how can we do that by genetic modification of the pig? Well, once we have that information, then we can build our plasmids, do our genetic engineering, whether that's using these molecules to inject directly into freshly fertilized eggs or zygotes, and then doing embryo transfer to get offspring, or if that's doing a genetic modification in a somatic cell in the culture dish and selecting those few cells where that modification has occurred, and then use that in somatic cell nuclear transfer to basically clone that animal from the cell. So somatic cell nuclear transfer. Take-home message, guys, is DNA makes RNA makes protein. We change the DNA sequence, we're going to change the RNA sequence, we're going to change the sequence of amino acids in those proteins. And when you change the sequence of those amino acids, you're going to change the three-dimensional structure of that protein so that it has a different function. So I want you to think of a hammer, okay? Think of a hammer and think of it as something within the cell, a component within the cell. You've got two parts. You've got a head and you've got a handle. Now, if I was a geneticist and I discovered this, I would say, well, that's nail driver 1 and nail driver 2. The head's number 1, the handle's number 2. Well, then Jason Ross is studying this same protein And he says, Prather, this thing doesn't drive nails, it pulls nails. Because the hammer also pulls nails. Well, you know, I can make it drive nails better. Maybe I make the handle longer by adding more amino acids. Maybe I make it fatter by changing the sequence of amino acids. Maybe I make the head bigger so it drives those nails harder. Maybe what I really want to do is pull those nails better, and I'm going to make the claws different. Maybe I'm going to put 2 heads on the hammer. Everything that I just described, if that was a component in the cell, a couple proteins, as long as they fit together and as long as the hammer functioned the way I wanted it to, that's within the realm of possibility. I want you guys to think of a cell as a factory, okay? And this factory replicates itself. It takes in raw materials, carbohydrates, amino acids, other things, just like any other factory takes in steel and wood and whatever. And it not only makes the tools to make the parts, to put together to make the machines, to make the tools, to make the parts, to make the factory so that it can make another factory. And all the blueprints for that are in the DNA. All the blueprints that are there. And if you want to swap out a page in those blueprints, if you want to get rid of a page in those blueprints, if you want to change a few letters in that blueprint to change what's made, you can change it. So I'm going to say a little bit about this new thing you guys have probably heard about, CRISPR Cas9 gene editing. And basically what this is the swine genome has about 3 billion letters. And if we take a unique sequence of about 20 letters, we can design this system so it'll recognize that, and it will go in and find that in the genome, and it will cut it. Now, the cell has lots of mechanisms to fix DNA damage, and what it does is it'll fix it. But the CRISPR-Cas9 system's still there. It'll cut it again. And the cell repair mechanisms will fix it. And it'll keep going through this cycle until the cell repair mechanisms make a mistake and it inserts a few letters of the genome, or it takes away a few letters of the genome. That's what non-homologous end-joining is. So you get additions or deletions of a few letters of the genome. So you can change a few bases, the letters of the genome. So you can maybe take out a piece. Maybe you want that hammer shorter and you want to pull out one section of that hammer handle. You can pull that out by deleting a section that comes out of the middle. Or you can just get rid of the hammer altogether. Get rid of the handle or get rid of the head so that it won't function. How long do some of these things take? Well, if you're going to engineer some of the somatic cells, some of these projects take quite a while. I'll give an example later, but it can take a month to a few years. If you're going to be able to do zygote injection, you can design these CRISPRs overnight, basically, and build them the next day, or order them from IDT, and they'll show up, and then you use them. And the somatic cell nuclear transfer, the zygote injection, it takes about a week to do the way we do it anyway. We go to the slaughterhouse and get ovaries and bring them back to the lab and mature the eggs, immature eggs in the lab, mature them so they're ready to be fertilized. We fertilize them, and then we have zygotes that we can work with. Gestation, 114, probably push 116 days. Puberty, 6 to 9 months. Okay, with that background, let's talk about PERS, and this audience knows about PERS. First detected in the US in '87, described in Europe in 1990. Type 1, they described it first, so they named the European strains Type 1. The North American strains are Type 2, and that's important. That distinction is going to be important later in my talk. But the virus replicates in alveolar macrophages, or white blood cells in the lungs. It also predisposes pigs to other bacterial and viral pathogens, partially because macrophages that clean up some of these things are gone. And you guys know that the vaccines don't work very well. You guys can read through that list. The bottom line is these pigs still get sick. What does it cost? Well, it's been estimated about $664 million a year in North America. That's 1.5 billion euros in Europe. Guys, that was $6 million yesterday. That's $6 million today. That's $6 million tomorrow. And that's just North America and Europe. That doesn't include Asia and the rest of the world. We can sell this all we want on the economics, and the economics are there for us. But that's not the way we're going to sell it. We heard this morning, we've got to put some emotion into this. Guys, this is a sustainability issue. We can't afford to be putting inputs into animals that underperform. Guys, this is a threat to our food security. We can't have animals dying and not have food to eat. This is an animal welfare. Who wants to see sick and dying animals? Nobody wants to see sick and dying animals. Then there's a psychological and emotional cost, and I think this is one of the things that really gets overlooked in this. When I talk to producer groups, and I get to this point, I talk about, okay, mom and pop producer, they're sitting at the kitchen table at night, and one of their barns broke with PRRS. They're trying to decide how they're going to pay the bills, or if they're going to be able to pay the bills. They're trying to decide if they're going to be able to send the kids to college. And you think about the psychological and emotional interactions between, in many cases, a husband and wife, and the stress that puts on it. There's not enough stress in life already, but the stress that then adds to it. You know, sometimes when I describe this to producer groups and there's a husband and wife sitting together, the husband, he always stares stoically ahead. And the wife, she'll kind of tip her head to the side and grab his shoulder and start to tear up. Guys, this is not a faceless corporation that this disease affects. Yeah, it does affect corporations, but this affects producers. This affects families. It's not some faceless corporation. And this is the only way we're going to sell it to the consumer, that we should be allowed to use this gene editing technology that I'm going to talk about to cure this disease or prevent this disease. So what does the PRRS virus do? Well, there's a model presented a number of years ago by Van Breeden where the virus would first interact with some molecules sticking up on the cell surface called heparan sulfate, siloadhesin, which is also called CD1 or Siglec-1, and then also CD163. And so what they proposed is that the virus would first come in and have a loose docking with this heparan sulfate and then attach to siloadhesin. That would then get internalized into a vesicle within that macrophage, within that white blood cell. The pH would drop, and then CD163, specifically domain 5 of that molecule, would then uncoat the virus so that the viral genome could be released and infect the cell. Okay, that's a model. We can test that. So the first thing we did was try to knock out siloadhesin. And these were our pigs born. They were born in 2011. And these pigs had one allele, one copy either from, you got two copies of every gene, one from mom and one from dad. Well, we had disrupted the sequence in one of those copies. But they're all boys. So we had to breed them, get some offspring, some brother-sister mating, so we could get animals that actually had both alleles disrupted. And this was our first litter. Mendel was very kind to us. We had 4 that were knockouts, where both alleles were disrupted, so that there was no siloadhesin sticking up on the cell surface. It just wasn't there. We had 4 heterozygotes, so they still had a good copy, so they still had siloadhesin sticking up on the cell surface. And then we had 4 wild-type animals that, they have both copies sticking up on the cell surface. Well, we shipped these pigs to Kansas State University because Bob Rowland at Kansas State University basically has a factory where he's taken in genetics from all over the United States and tried to look for natural resistance to this PRRS virus. He hadn't found any. Well, maybe a little bit of genetic response, but hasn't found any animals that are resistant to it. So we shipped these pigs to Kansas State, and basically the short story is that, first of all, the siloadhesin mutation, because we don't know this on every mutation, it's not embryonic lethal. Okay? You don't know till you make it, so it is compatible with the life, but the pigs were still susceptible to PRRS, so that was kind of disappointing. If anybody has a use for these pigs, they're going really cheap right now. So we conceived the idea in '02, and we looked for people to help us do it in terms of funding. And we plugged along, and finally, we made some progress, and we had our pigs born in January of 11. The publication was in 13. That was 11 years from the time we came up with the idea and started moving down that road before we got that project out. That was the longest project I've ever been on. And now things have changed in terms of being able to do these genetic modifications with CRISPRs. It's just a day and night. We can move light year speed now. So I want to back up. There's a whole series of experiments that implicated siloadhesin, so that's why we started with that one. But there's another series of experiments that implicated the CD163 molecule. And there's other candidates out there, CD151, it's a tetraspanin, a vimentin, a DC sign, which is also called CD209. Anyway, there was this debate, well, which molecule is really important? And so there was another paper by Van Gorp where they took cells and they put in a gene for siloadhesion and a gene for CD163. And then these cells could get infected in vitro, so they could study them in the lab. And then they said, okay, well, what about if we change parts of CD163? Can the cells still get infected? And so some of the things they did is so this is going to be our pig CD163. It has these scavenger receptor cysteine-rich domains. It's a repeat of about 9 domains that are really quite similar. It's like beads on a string, kind of. And then I also have human CD163 here in gray, and then another molecule called human CD163-like protein that actually has the same domains, but they're numbered differently. So this is numbered domain 8, but it's the same as domain 5, basically. And what they did was put these different molecules into these cells, and they found out when they put in the human CD163-like that the cells didn't get infected anymore. So this is what part of that is really involved. And so they did some experiments where they swapped out domain 4, 5, and 6 with 7, 8, and 9. And they found out that those cells didn't get infected either. And then they looked at 7, 8, and 9 individually, and they found out that when they swapped out 5 with 8 that the cells didn't get infected. And so we thought, well, this is something we want to move forward with. And so we wanted to do 2 different things. One is we wanted to knock the gene out, make it so that it didn't make any protein at all. And then we also wanted to do that same domain swap so that we would have a CD163 molecule on the cell surface just altered a little bit. And so why did we want to do that? Well, those other domains were thought to be important for life. Okay? One of the functions, domain 3, is thought to be involved in haptin hemoglobin clearance. In fact, these CD163 knockout pigs do have higher haptoglobin levels circulating in their body. Domain 2 was thought to be involved in erythroblast blinding. 1 through 4 and 6 through 9 were a member of the tumor necrosis factor, superfamily 12. Bacteria are thought to be scavenged partially by number 2. We kind of want to keep that. It was thought that number 5 was for PRRS. There was some evidence out there that number 2 was for African swine fever. Well, we've taken our knockout pigs and now challenged them with African swine fever. They get sick and die just like wild-type pigs. So that's not involved. So we did a couple different things. We used the CRISPR Cas9 system to edit our donor cells so we could do that domain swap. And then we also used this same system, we injected it directly into zygotes to try to just knock out the gene, disrupt the sequence so it didn't produce any protein at all. And one of the ways was somatic cell nuclear transfer. Basically, we take unfertilized eggs, we insert a micropipette, we pull out the chromosomes, and then we replace it with our donor cell that has that domain swap where we've swapped that domain 5 with domain 8. We also do zygote injection. In this case, right after fertilization, we will take the CRISPR Cas9 system and we will inject it directly into the 1-cell stage embryo, the zygote, and then do an embryo transfer. So again, the timeline, the first part of this you've already seen, but in the case of CD163, from the moment we instituted the CRISPR Cas9 system, we had our pigs in 6 months. Okay? This just changed the way we did everything. So we made this domain swap targeting vector. I'm not going to go into details on this. For those of you that are genetic engineers, I'll be glad to, but I think for right now we'll just kind of skip through this and focus on this. We screened over 3,000 colonies looking for this domain swap and couldn't find a thing. And the first time we tried CRISPRs on it, we had our cells. Just worked like that. It was just, wow, this technology is so useful. So our timeline, and these are the dates, this is July of 13. We had our pigs by January 13th and 14, 5.5 to 6 months from the time we implemented the technology till we had the pigs. So what I'm going to talk to you about first is a knockout animal where normally you'd have CD163 sticking up on the cell membrane. The first animals I'm going to talk about, there's no CD163 that sticks up on the cell membrane, and I'm going to talk about the wild type animals, or an animal with a normal sequence of DNA making a normal CD163. So here's the pigs we made where we had knockout animals, and this is right before we shipped them over to Kansas State. I drove them over there and left them. Can you tell who's the knockout pig? I don't think you can. These pigs look perfectly normal. They grow fine, they reproduce fine. They don't get sick more than any other pig. We raise them in a pig barn, basically, And they're exposed to bacteria and pathogens just like every other pig in our facility. They're doing fine. But you can't tell which pig's which. So that pig in the front is actually number 40, and it has a 7 base pair addition in exon 7. So we added 7 letters into the genome within that coding region, and basically it disrupted the sequence so that it didn't even make the protein. The other allele, that was the one this pig inherited from mom. The one it inherited from dad, it had 11 letters, 11 bases, actually introduced into that region, made it a little bit bigger, and disrupted the sequence again, so that it didn't make any protein. So in that pig, we changed 18 letters out of 3 billion. 18 letters out of 3 billion. I drove the pigs over to Kansas State, left them, drove back home. Bob Rowland has his factory there that does this type of challenge. The crew at Kansas State, they didn't know which pigs were which. They had ear tags. We didn't tell them any ear notch numbers or anything. They were allowed to acclimate. They use this virus called NVSL. They call it a relatively hot in terms of replication and pathogenesis. The pigs were all in the same pen. They introduced this virus both intranasally and intramuscularly. After 35 days, the study was terminated, and the lungs and tissues removed for pathology. So I drive back home. Bob Rowland leaves, and he goes down to Florida. And from the beach in Florida, I get this email. Randy, below are the results. Everyone busted their butts to get it done. Let us know if we got it right. And this is from Maureen Kerrigan, his technician in the lab. Pigs 40, 43, and 55 remained negative 4 days post-infection. They had never seen this before. In fact, when the technician came in with the data and gave it to Maureen, he didn't know what he's doing. In fact, he was saying, I've never seen this before. This has got to be messed up. We've never had any pigs negative. It's got to be something with the assay. Maureen said, it's fine. Let's just go on to the next time point, and we'll come back later and deal with it. So the people in the lab running the assays, they were blinded to it. They didn't know what was going on. So basically, about half the pigs in the wild type pigs, about half of them got respiratory signs after about 7 days. About half of them got a fever after about a week. The 3 knockout animals, the null animals, no respiratory signs, no coughing. They're in a pen with other pigs coughing all over them. No coughing, no respiratory signs. So this is the typical response that they get when they measure the amount of virus in the blood. And it comes up on day 4. It comes up real high. Guys, this is a log scale. So when you see a 6, that's like a 1 with 6 zeros. This is a huge amount of virus that shows up in the blood of these animals. And by day 4, it's huge. And then it tapers down, and that's a pretty normal response. The guys at Kansas State have challenged over 3,000 pigs, and they see this every time. Every time they see this. Well, what did they see with our pigs? So these are the wild-type pigs. They come up with a pretty good viremic response like you'd see. The knockout pigs that don't have any CD163, they're down here in the triangles. These pigs had no virus replicating within them. In fact, you look at the antibody production, these pigs, the wild-type pigs, antibody production comes up about day 14. The knockout pigs, they didn't see enough virus to even develop an antibody against it, at least a detectable response. It's like you were measuring my blood and looking for viral replication in me because I was in the same room with the pigs. There's nothing there. For those of you clinicians, this is a wild-type animal and showing all the pneumonia and mononuclear cells that have infiltrated the lung of this pig, this wild-type pig. The knockout pigs, the lungs look perfectly normal. So there's a couple studies out there that support this that have been done in vitro only. In other words, in the lab. They haven't really infected it in pig. And this is a study where they said that the 561st amino acid in the sequence of CD163 is an arginine. And they say if we mess with that, we can change how the virus then infects those cells. Another study by the Roslin group basically said if we take domain 5 out, so now it goes from domain 4 to 6, and just kind of splice those together so that molecule, that whole chain is a little bit shorter, those cells don't get infected either. Okay, we talked about the knockout animal. We talked about the wild-type animal. Now, what about the domain swap, where we've swapped domain 8 with domain 5? So this is a table showing the data that we generated. So we took pigs that had wild-type, had the domain swap, and had a knockout. And we took cells from those animals and we challenged them with a whole bunch of European strains up here at the top and a whole bunch of North American strains. And what you see with the wild type animals, and somewhere around 50% of the cells get infected. So 50% of the cells get infected, that means the pigs get sick. If we look at the knockout animals, the null animals, the cells from the null animals, none of those cells get infected at all from either types of these viruses. So there's no replication of the virus in these cells, that they don't get infected. Well, what about when we did the domain swap? Now, remember I told you that that previous study was done where they did the domain swap and they said they didn't get infectivity. Those were done with European strains of the virus. And that's what we have here at the top. And you see that 0% of those cells got infected with the European strains of the virus. In contrast, the North American strains of the virus infected anywhere from 3 to 12% of the cells. So the question is, if 3 to 12% of the cells get infected, does that make a sick pig? Well, the answer is yes. So here we have our type 1 viruses on the left, and this is a challenge. So this is the amount of virus in the pig's blood. Over here we have wild type animal. The viral load comes up like normal. The knockout animals are down here. So are those domain swap animals. So the European strains don't infect the domain swap animals. In contrast, on the right, we have our wild type animals up here with our North American strains, our knockout animals down here, and then we have the boxes. And we have one animal that came up a little bit late. The rest of the animals came up pretty much with the wild type animals. And so that's telling me that, okay, even if you have a small percentage of the cells get infected, the pigs still get sick. And the only way to stop this virus is to stop it from infecting the cells. Okay, what about reproductive PRRS? We talked about the baby pigs. What about an animal that's pregnant? So how does a fetus get infected? So there's reproductive PRRS that accounts for about 45% of the losses that people see with PRRS. It results from infection about day 90 to 114. Again, it replicates in the maternal or mom's macrophages, mom's white blood cells. The virus then crosses the placenta to infect individual fetuses. And it's interesting, you can have fetuses right next to each other where one's infected and the other one's not. And there appears also to be some fetus-to-fetus transmission. And the primary site of infection in the fetus is a little bit different. It appears to be in the thymus. So we concocted an experiment where we had wild-type pigs, knockout pigs, knockout sows carrying knockout fetuses. We had wild-type sows carrying wild-type fetuses, so they should get sick, the fetus should get infected. This animal should not get sick, the fetus should not get infected. But we wanted to know if a knockout sow could protect a heterozygote fetus. So a heterozygote fetus still has one good copy, should still be susceptible to infection. But if mom doesn't get sick, even though she's been inoculated, is the fetus going to get sick? So again, basically the same trial we described earlier, that we use this NVSL 97, half administered intramuscularly, half administered intranasally. The pigs are co-housed, so when one starts coughing, it's coughing on everybody. The CD163 knockout gilts, they had no clinical signs, just like we saw previously with the smaller pigs. The wild type animals, one aborted on day 106, and the viremia was a 5.5, so it was really high. And then day 17 to 19, we euthanized the animals. So this slide's really pretty busy, but let me walk you through it. So we have mom's number 138, and number 140. We have her viremic level at the end of the study, so she was about 3.6, 4.1. They were wild type, the fetus was wild type, and this is her right uterine horn and her left uterine horn, and we've numbered the fetuses from 1 to 16. And if we look at the numbers within the circle, that describes their morphology. So a 1 is a normal morphology, out to a 5 is dead and necrotic. So we have dead and necrotic fetuses next to perfectly normal fetuses. We also have numbers down here that indicate the amount of virus load within that fetus. And so you can have an animal that has a non-detectable virus load between two animals that have a very high virus load, or two fetuses that have a very high virus load. So this is what you would normally see with the wild type animals. Well, what happens when you have a knockout mom carrying fetuses that should be susceptible? And here's what happens. They're normal. They have normal morphology. They aren't carrying any viral load, just like mom's not carrying any viral load. And in contrast, or similarly, if you have a knockout mom carrying knockout fetuses, again, the fetuses look perfectly normal. There's no sign of infection. Okay, so most of the wild-type fetuses from the wild-type gilts were infected, positive for PRRS. There's wide variation between fetuses. Viremia is not always correlated with gross pathology. If we would have gone back and looked at fetus number 5 from litter 138, had a very high viremic load, but really looked normal. And none of the pigs, none of the fetuses in CD163 knockout gilts were infected, and the morphology was normal. So what are some of the conclusions for the PRRS part? Well, siloadhesion pigs are not resistant to type 2. We haven't really tested them with type 1. We're going to do that when we get a chance to collect some more cells, at least test it in vitro. The CD163 knockout pigs are resistant to both type 1 and type 2 viruses. The CD163 knockout pigs provide protection to fetuses in utero. And we know that domain 5 is critically important in the different types of viruses and how they use domain 5 to infect the cells. So we have the model out there that says it binds to heparin sulfate, then gets to siloadhesion, gets internalized, and then gets uncoded by CD163. Well, we tested the siloadhesion part, and the pig still got sick, so that's probably not part of the model. But CD163 appears to be a gatekeeper. Now, I'm not going to criticize the people that came up with the model because they came up with an idea how it might work. And I encourage that because now we have the tools that we can test those models and determine, is that really the way the virus infects the cell? We can change any molecule in the cell you want. So one of the questions I often get asked is, well, knockouts are bad, aren't they? Aren't they all lethal? We can't have any knockouts. Well, there are some that are embryonic lethal. There are some that are neonatal lethal. We made cystic fibrosis pigs. That's the CFTR for the group over, well, across the way at Iowa, University of Iowa. And this is the only model people have to study cystic fibrosis that replicates what happens in humans except for kids. Incredibly valuable tool for the physicians to come up with treatments and therapies to treat these people. It's neonatal lethal if you don't treat it. These two sets of pigs are pigs that we've made that have a compromised immune system. Yeah, that's going to be lethal once they need their own immune system to live. There's other mutations. Duchenne's muscular dystrophy, Jason Ross back here, made that. It's not lethal. It can be detrimental. We have pigs that get phenylketonuria. It's not lethal. It's detrimental. We have pigs that have an APC mutation. This results in polyps in humans, and it results in polyps, intestinal polyps, colon polyps in our pigs. Okay? That doesn't show up till later in life. It's not lethal. Some mutations you make are going to make them infertile, and there's a whole bunch that we've made, listed here at the bottom, that have no general impact. In fact, you wouldn't know the pig had the mutation unless you went in and looked for that mutation or you looked for the gene product that you've altered. So just because we knock out a gene doesn't make it lethal, doesn't make it bad. So where do we go from here with CD163? Well, first we have to show that there aren't any unintended consequences. Are these animals unhealthy? They appear to be fine. Do they grow fine? They appear to. Are they efficient at feed conversion? Well, we don't know yet. That we haven't done any studies to test that. There's probably a whole number of other things that need to be done. We're going to have FDA approval, unfortunately. And the Food and Drug Administration treats every different edit that you make as a different drug. And so it's treated as a new drug and goes through the same regulatory process as every other new drug. Even though every cell division results in about 30 changes in the genome, 30 letters are changed, they don't regulate every fertilization or every meiosis. And those are random mutations. We don't know if they're bad or good. We know what we're doing with this. So what other things can we do in production agriculture? So I talked about disease resistance with CD163, but African swine fever is a real concern in Europe. And there's a group at Roslyn that took a gene from warthog, REL-A, that has, I think, 3 different amino acids than the REL-A gene in domestic pigs. And they made that change. They basically introgressed the warthog version to the domestic pig because the warthog gets infected with African swine fever, but they don't get sick. Can we make pigs that don't get sick to African swine fever? Bovine respiratory disease, CD18. We can change a single amino acid in our cattle, and we can make cattle that don't get the leukotoxin from Mannheimia hemolytica doesn't bind, and the cattle won't get respiratory disease. We can change a single amino acid. We have some pigs where we have a TMPRSS2 mutation, and that's a serine protease that's thought to uncoat a lot of these viruses so that they can then infect the cell. I'm really excited about moving forward with some of those tests. What about animal welfare issues? A group has taken, Recombinetics has taken the Celtic version of the polled allele and put it into high-producing dairy cattle. That's an animal welfare issue. Is that unhealthy to eat? It's already in Angus. And we're going to regulate that as a new drug? We have the potential to alter the productivity. We have the potential to improve the composition of the carcass. This is an incredibly valuable tool that we have that will allow us to answer a lot of basic and applied questions, both about human medicine, animal biology, and to improve production agriculture. So I'm going to acknowledge the people that have been involved really with the PRRS side of the project. Kevin Wells and Kristen Whitworth really pushed forward the CRISPRs. This is Kehoe Lee. He did some of the first microinjections with the CRISPRs into zygotes. We have a team that does our nuclear transfer and embryo transfer, another team of people that helps care for the baby piglets. John Green really pushed forward the silo adhesion part of the project. Bob Rowland and his group at Kansas State, some of our funding people at Genus, and then also the Christopher Columbus Foundation, Food for the 21st Century, and USDA, ARS helped fund a little bit of what I talked about today. I'll entertain questions. Thank you.
Thank you, Dr. Prather. Questions?
Do you think we're going to have to come up with some different technology to describe these kind of procedures to make it more palatable to the general public? Because whenever you start talking, GMO and genetic engineering, people just freak out. And This is not that. This is editing and deleting genes, not adding. And I understand all that, but most people are not.
Yeah, and they're not going to. And so you're going to have to talk about things like, this is sustainable. This is going to make it sustainable. This is addressing an animal welfare issue. These pigs are getting sick and dying. Do you really want us to, you want to prevent us from keeping these pigs from getting sick and dying? I mean, I think that's the way it's going to have to be sold. You can use the term gene introgression, and I think that's what the group at Roslyn used for introgressing the warthog version onto the domestic animal version. But yeah, you're
something though that's catchy and simple.
Well, so do it. I mean, I don't have the answer. I don't think there is the answer, because I think if you come up with something, you're lying.
I don't think you're lying. I think it's just like you said, you've got
but it is genetically modified. It's gene edited. It is edited. I'm on your side, but I'm saying what they're going to say is if you come up with a different name for the same thing that they see as the same thing, they're going to see that they can't trust us.
Dr. Prather, can you describe how the intellectual property is owned here, and do we have adequate laws that govern what's going on here, et cetera?
So the only thing I can address is CD 163. The University of Missouri has filed for intellectual property protection on that, and they have licensed that to Genus. But you've got, this is one disease, guys. This is a big sandbox. There's all kinds of things out there that we can do. This is some of the low-hanging fruit. And this shows what potential it has. You know, it's interesting talking to Bob Rowland. He says, when we first started this, he says, Randy, I need 15 or 20 pigs with this genetic modification to test it. And now Bob says, Randy, I need one. Because if they're not going to get it, they're not going to get it. So that sped it up quite a bit.
Two questions. As you mentioned, in every cell cycle replication, there are about 30 layers can change. Do you expect after several multiplication that this is going to revert to the original?
So would this revert? We're about 4 generations in and we haven't seen it. So science can't prove things don't happen. Science can only prove things do happen. We haven't seen it. I'm not going to say it can't happen because who knows?
And the other question is, if you show that the type 2 first didn't, or in the other way, when you swipe the domain 5, type 2 is still being pathogenic. So do you think that is because of the virus structure or because the virus is targeting a different domain?
So I think the virus is targeting the same domain, the same region of that CD163, but the virus is mutated a little bit so that it targets a little bit different part of that same molecule on CD163. Guys, structure is function. I think somebody said that this morning. Structure's function. We change the DNA sequence, we change the amino acid sequence, we put in different amino acids or change the sequence of amino acids. You change the three-dimensional structure of this protein within the cell, and now it's going to have a different function. Think of the cell as this factory where everything has a specific shape for a reason. And if you change the shape, it's going to cause it to do something different. And so think of this virus, picture in your mind this three-dimensional virus coming in and binding to CD163, that domain 5, and the type 1s bind here, and maybe the type 2s bind here, but they're still using that same piece of CD163 to infect the pig cell.
That's what scares people.
Why does it scare people?
Because it's kind of an unknown.
Yeah. Yeah.
So, as you said, bug rolling infected more pigs than anyone else. We've heard and haven't seen any natural occurrence of a resistant bug, right? And you give the example of the full cow, that you know that this gene exists in other breeds. So you're changing to something that you know that exists. So do you think that the first resistant gene is the same example for the whole gene? Because we haven't seen a natural occurrence of fully resistant.
So that's a better question for Jack Deckers, who's sitting back here. Is there a natural mutation that occurs in CD163 that's in anybody's database? And I think the answer is probably not.
Yeah, we haven't. Yeah, so we work with that data from both yeah, we haven't seen it. But there's lots of different breeds out there all over the world that we haven't looked at, and there are stories that some breeds are more resistant. Now, I haven't heard anybody say that they're completely resistant.
Yeah.
So my question is, do you think that these are the two? Are these comparable? So gene editing for something that we know that exists?
Well, I think the only way you can go with that is, in theory, there can be a natural mutation that occurs in CD163 to knock it out. And there might be one out in nature somewhere already. It's just that there's another good copy next to it, and so it's still making CD163, and it's still getting infected. You just haven't inbred it, so you have both alleles with that edit and tested it for PERS resistance.
I think for the sake of time, we're going to go ahead and move on, but let's put our hands together for Dr. Prather.